Transfusion medicine is the essential science of managing the human immune system's complex response to foreign blood products. The clinical stakes are absolute. Transfusing an incompatible blood type directly triggers a fatal hemolytic reaction. When this happens, the recipient's immune system identifies the donor's red blood cells as a hostile biological threat and violently destroys them. This image shows the two distinct physical components of whole blood that drive this conflict. At the bottom is the dense layer of solid red blood cells, and at the top is the liquid yellow plasma. The solid red blood cells carry specific surface markers. They are the targets. The liquid plasma, meanwhile, carries the antibodies, the immune system's weapons. Patient survival relies entirely on flawlessly mapping the biochemical reciprocity between donor cells and recipient plasma before a single drop of blood is ever transfused. The primary non-negotiable rule of the ABO system is that ABO antibodies are non-red blood cell stimulated. This means humans natively and automatically produce plasma antibodies against any blood antigen they do not personally possess. Starting with a biological baseline red blood cell without surface markers, we map blood type A by placing structural antigens onto the cell. In reciprocal immune response, the surrounding plasma automatically fills with circulating anti-B antibodies. The inverse is true for type B. The cell has B antigens, and the plasma holds anti-A antibodies. For type AB, the cell carries them with A and B antigens, meaning the plasma contains 0 antibodies, which prevents the blood from attacking itself. This chart details the full matrix, but the far right group O column reveals a unique anomaly. The cell surface is completely blank, yet it is paired with a highly aggressive plasma profile containing both anti-A and anti-B antibodies. Because this system operates on absolute biochemical reciprocity, an adverse reaction is a mathematical certainty if the wrong variables are mixed. Applying this ABO logic to clinical reality dictates how we safely route donor resources to recipients. This grid tracks compatibility. Notice the vertical O-negative donor column. Compatibility drops fill every single recipient row. Type O is universally accepted for red blood cell transfusions because its blank surface offers 0 targets for a recipient's immune system. A clinical flip occurs when we stop transfusing whole red blood cells and instead transfuse the liquid plasma fraction. The rules invert. The strict rule of plasma compatibility is that plasma-containing antibodies cannot be given to a patient who possesses the corresponding cellular antigens. This table maps that plasma logic. Looking at the bottom AB row, you see yes indicators across the board, identifying type AB as the universal plasma donor. Type O is the universal red blood cell donor because it lacks the targets for the immune system to attack. Type AB is the universal plasma donor because it lacks the weapons to execute an attack. There is a secondary required system for compatibility mapping, the Rh factor, specifically the D antigen protein. This illustration shows the structural difference. The Rh-positive red blood cell is studded with proteins, while the Rh-negative cell is completely smooth. Rh compatibility operates on a different biological timeline than the ABO system. A patient who is Rh-negative does not automatically possess native anti-D antibodies in their plasma. Anti-D antibodies only manifest after a specific sensitization trigger. The patient must be physically exposed to the foreign D antigen through a blood transfusion or pregnancy. This diagram illustrates hemolytic disease of the fetus and newborn, the most severe consequence of this exposure rule. Notice the anatomical separation here, showing the Rh-negative maternal space surrounding an Rh-positive fetal space. During an initial exposure event, typically childbirth, fetal Rh-positive cells breach the maternal space. This permanently sensitizes the mother's immune system to the D antigen, prompting it to generate anti-D antibodies. In a subsequent pregnancy, the mechanism is already armed. The newly established maternal antibodies cross the placenta to attack and lyse the fetal red cells. An ABO mismatch kills a patient immediately. An Rh mismatch creates a delayed immunological time bomb that strictly limits all future medical options. These theoretical models are proven empirically on the benchtop using specific laboratory diagnostics to authorize a transfusion. This graph charts agglutination, the visible clumping of cells in a test tube. In the equivalent zone, you see the physical lattice formation of antibodies locking multiple red blood cells together. This acts as our diagnostic proxy for an in vivo hemolytic reaction. As shown in this diagram, the first step of verification is forward grouping. Standardized lab anti-sera, the blue anti-A and yellow anti-B, are dispensed directly into the patient's red blood cell samples. We read this matrix to confirm the presence of antigens. Looking at the upper left quadrant, a severe 4+ agglutination in the anti-A column confirms the patient has group A blood. This is followed by a required secondary verification, reverse grouping. Here, we test the patient's plasma against known A1 and B lab cells to map exactly which antibodies are circulating. Safe transfusion medicine demands dual empirical proof. We prove exactly which antibodies exist on the cells and exactly which antibodies patrol the plasma. In a severe trauma scenario, a massive hemorrhage patient arrives bleeding out, and there is zero time to perform forward or reverse laboratory typing. This grid displays the diagnostic complexity required for a standard crossmatch. Running these tests takes time that an actively bleeding patient simply does not have. To save them, physicians rely on the Universal Trauma Failsafe Protocol, administering uncrossmatched Group O blood to bypass ABO compatibility testing entirely. The RH sensitization rules we established earlier dictate exactly how this emergency blood inventory is managed. To preserve scarce resources, all males and non-childbearing females receive O-positive uncross-matched blood. But there is a critical exception. Females of childbearing age and pediatric patients strictly receive O-negative blood to entirely eliminate the risk of inducing hemolytic disease of the fetus and newborn later in life. Internalizing the precise, predictive mechanics of cellular biology allows clinicians to save lives under the most extreme pressure.